रिकॉर्डिंग शुरू हो गया है। स्टार्ट। दो मिनट ठहर जाओ, चल भी रहा नहीं चल रहा मान रही। Today's lecture is now replication, the replication of the virus. How the replication of the virus take place. And they have different steps. And the word replication means the action of copying or reproducing something. Okay. Now, after the viral genome has been uncoded, transcription, translations of the viral genome is initiated. In order to understand this, there is a viral growth curve in which the viral replication is often analyzed by the growth curve. In this viral growth curve, the viral multiplication is present. Host cells is measured as a function time. Now there are four phases in the viral growth curve. Number one is inoculation. Number two, active phase, which is penetration. The number three is exponential phase, which is also known as the latent phase, and fourth one is the stationary phase. In case of inoculation, the virus come in contact with the cell, and it landed over the surface of the cell and attach itself with the membrane of the cell. So after the attachment, the viruses, they penetrate their genetic material into the cell. The genetic material, when enter into the cells, it penetrates into the cells, and after the penetrations, there's uncoding of the viruses. Uncoding of the viruses take place. So every virus, every part of the virus has been dismantled. Uncoding happens inside the cells, where everything has been scattered. The capsule has been scattered. The genetic material has been scattered, and the new and new genetic material and the proteins are found in the cell, from which these materials assembling take place inside the cell. After the assembly means the new progeny viruses has been formed in multiple amount, a huge amount, and hence they are released after budding or lysis of the cell. The elect phase, in case of eclipse phase, is a penetration. It is a time between the injection or penetration and appearance of the new phage. It is the viral progeny formations are formed, called as eclipse phase. And the third one is exponential phase, which is related to the log phase. It's a time between the injection and the rupture of a cell wall is called the latent periods. And so the final phase, the fourth phase, is the stationary phase, in which the production of the viruses comes to slow down and ultimately ceases. The rate of cell division and the death roughly equal. On the other hand, the case of complete viral replication, there are 8 steps which which occurs in the replication of the complete virus formations. Number one step is the adoption, that is the attachment of the virus on the surface of the cell. Second is then after the attachment, the penetration, endocytosis is formed. The surface of the endocytosis is formed on the surface of the virus, and through this endocytosis, penetration of the genetic material into the cell. The third one, after the penetration, There is uncoding of the genetic materials of the whole virus. The uncoding of the whole virus. The capsid have been separated, the viral genome have been separated, and they take out the replication machinery of the cell in their own control. The cells take the replication machinery in control. So there is transcription and translations occurs. Transcription means the transcription of the viral genome into the messenger RNA. The whatever genetic material is, either RNA or DNA, has been injected, they should be transcribed into messenger RNA. The DNA of the genetic material of the viruses are directly transcribed into the messenger RNA with the help of viral enzymes. On the other hand, the RNA, the RNA material, genetic material there, then there is then there is then this RNA is transcribed into first DNA, and then this DNA will be translated into messenger RNA. And hence, the new genetic material proteins have been formed inside the cells, and finally, the assembly of the material will take place. New progeny viruses are formed inside the cell, and they are released after the after the lysis or budding of the cell takes place. In this way, the whole 8 steps take place in the replication of the viruses. So these are all cells I am showing you now, which I already explained you. See, there is the blue line is a cell, and there is a landing of the viruses on the surface of the cell. Then there is endocytosis and attachment and endocytosis, and then they enter into the cell, and uncoding is taking place, and transcription, translations take place, protein formations take place, and assembly of the virus is taking place, and new progeny cells are formed, viruses are formed, and they are released after bursting or lysis of the cell. These are all endocytosis, which I already explained you, and coding and assembly, and the same thing. They just explain you. Now there's a life cycle of the bacteriophage. In the life cycle of the bacteriophage, there are 2 cycles. One is the lytic cycle, and another one is the lysogenic cycle. And this diagram shows you is the diagram of a bacteriophage. So in case of lytic cycle, which is also known as master and slave relation, where the virus is the master and the slave is the bacteria. It is a phage replicate cycle which results in the death of the host cell. In the lytic cycle, there is death of the host cell. There is a burst and death of the host cell. While in lysogen cycle, there is no death of the host cell, but there is integration of the genetic material of the viruses into the cell genetic material. Integration. No lysis or burst or after in the lysis. It only happens in lytic cycles. The lytic cycle, the multiplication process of virulent phage is called lytic cycle, because the host bacterium is lysed at the end. It is commonly referred as a reproductive cycle of the bacterium. Now the steps of the lytic cycle are 5. There is attachment of the viruses on the surface of the cell. Then there is endocytosis and penetration of the genetic material of the virus into the cell. Then there is biosynthesis, transcription, and translation. They take place. Hence, the assembly of the cell with genetic material, and it takes place, and the progenies are formed there. Ultimately, there is a release of the lysis. The release or lysis of the cell takes place. In this way, the progenies are formed, and they are liberated to invade the other cells, healthy cells, and this replication goes on. You all, which I already explained just now, or is bacteriophage. Now is the lytic cycle, and the diagram of our lytic cycle and lysogenic cycle. Well, there are 2 cycles. This way, This figure is showing two cycles. One upper one is the lytic cycle, and the lower one is the lysogenic cycle. This is the lytic cycle. This is the lytic cycle. It starts from here. Arrow indicated there. Okay. These bacteriophage, they are coming and they are landing on the surface of the cell. Now, after attaching, they're introducing, penetrating their genetic material. They may be either RNA or DNA. These bacteriophage, they are introducing the genetic material inside the cell. And this is the DNA or genetic material of the cell, the brown blue color. And this is a red color. The bacteriophage genetic material is a red color. So it is introduced into cell, and ultimately they are the viruses integrated. The virus genetic material. See, this red portion is integrated into the genetic material of the cell. So there is integration of the cell. There is no any uncoding of the cell taking place. Now here it will happen two things. Whether this cell or this condition of the cell in which there is integration of the virus cell goes like this and into this phase, there is uncoding of the genetic material take place, or this integration may be divided into two portions. The genetic material which is integrated here will be converted into two portions. The mitotic cell division will take place, and the mitotic cell division is taking place here, and ultimately further the mitotic cell division will divide this one into two portions like this. So one portion, other portion. In this way, the genetic material has been liberated, and the infection of the cell will go on taking place. If this integration is entered into this form, there is uncoding of the genetic material. The port has been integrated, and uncoding takes place. The genetic material of the plant, and ultimately there is formation of the capsid and other genetic material. The genetic material will here showing comes under the genetic capsid, and the genetic material under this capsid, and hence ultimately they form a tail after the genetic material of the capsid takes place. The tail is formed here like this. After the formation of this tail, the progenies will be formed inside the cell, and they are so much formed, they produce either pressure on the wall of the surface, and hence the lysis of the cell will take place, releasing the complete infected phages or the viruses. In this way, the lytic cycle will take place. Lytic cycle, remember, I hope you remember, lytic cycle, there is only bursting of the cell, destroying of the cell take place, and the progenies are ready. But in this case, there is no release of the genetic material, no lysis of the cell taking place here, and hence they are integrated here, in which the lysogenic cycle takes place. I mean, when the genetic material are integrated and they are not uncoded, this happens in the lysogenic. But when the integrated genetic material are formed inside the cell and they are integrated or uncoding of the genetic material take place after the integration, the progenies are formed in the lysis of the cell take place inside the cell. So this is all about the lysogenic cycle and the lytic cycle, which I already explained you just now. So this is all about the lytic cycle, which I already explained you. Thank you very much. खुदा हाफिज। अली एक तो हो गई, रेप्लिकेशन हो गया आपका।